about 90% of our diseases affect really small numbers of patients, sometimes referred to as ultra rare. So we just need to keep trying. Those are very difficult diseases for industry to step into, because we have diseases that have 10 patients. It's just really hard to make any kind of a business model around that. So some of the work NIH does is we're trying to develop efficiencies into how we do this, both with the drugs themselves or the preclinical information or even the clinical trials. And I think that's an area where these public sector or public-private partnerships can just be of tremendous benefit.
Our hope in some of the programs like the Bespoke Gene Therapy Consortium, the Ph.D. program and others is to make that time shorter and the expense less and the overall process more efficient, because essentially if you keep doing the same thing over and over again and you're just changing out the therapeutic gene, it should be possible to make the process more efficient and less costly. And even though gene therapy could be very effective for those diseases, and even though we have data in animals to show that it works, getting to the point of the clinical trial is really difficult because there is no commercial interest, and often it falls to the families of patients with these diseases to come up with the money and ultimately fund the clinical trial and bring it all the way to the point of getting the clinical trial started. And that's really a tough situation to put people in. What we're trying to do is make the whole process simpler and more streamlined for these diseases like that and the parents who are trying to bring them forward, so they can see sort of a straightforward template to get those clinical trials up and running really quickly. Because in some of these diseases, there may never be a marketed product. We may end up treating all the patients in the context of a clinical trial. And so we're trying to find a pathway and make that as efficient and easy and inexpensive as possible.
NIH is seeking to leverage commonalities across diseases, but also across some of the therapies or therapeutic approaches that we might try. We have anywhere between 7,000 and 10,000 rare diseases, and potentially even more than that. But there may be some commonalities, or we know that there are commonalities to some of the underpinnings of these diseases.
It's a platform because to make a new treatment for a new disease, you can simply swap out the therapeutic gene and thereby make different treatments for different diseases. I say simply, it's not really that simple, but at least in principle, it is straightforward to do and therefore is a therapeutic platform. And indeed, we've already seen that it's being used for multiple diseases, both in clinical trials, and we've even had 2 approved products. The idea is that if we can use the same platform, that could be applicable to other diseases that would be amenable to gene therapy using an AAV9 vector. That's really the whole goal of this, is to extend this platform as much as possible. Also, it's the learnings we're going to get from this are not limited to AAV9. The basic idea is if you use the same AAV serotype and all of the vectors are made in the same facility using the same process, the same purification, the same sort of efficiencies that we hope to demonstrate with the AAV9 should be applicable to other AAV serotypes as well. I think keeping the process the same for all of them is really important. Our colleagues at the FDA often say that the process is the product. And so, keeping that the same is, I think, a really key aspect of PAVE-GT.